Hi guys, welcome to topic 2 in chapter 9. This topic is all about physical controls and how we use these to control microbial growth. So why is this topic important? In hospitals or other clinical settings, you're going to use a lot of physical controls. So it's important to understand what they are and how they work and what their result can be. So it's really important to have a good handle on these. Just like I said before in the last topic, I expect you to have the details down for these. So make sure that you really focus in on this material as it is presented in the textbook and in this lecture. As you can see, we have a lot of objectives here, so this is going to help you master a lot of those concepts that we're talking about. So make sure you can get them down. One thing I want to point out is while we talk about 5 methods of control here, there's actually 6 because heat is divided into 2 different types of physical controls. So just be aware of that when you're looking at your slides. So as I said, heat is 2 different types of control, and heat focuses on the denaturation of proteins. And we talked about this a little bit in the last section, where the proteins, when they're exposed to heat, they unfold and they can't refold, which means they can't function anymore, which means the cell is no longer able to survive. So it's a very effective way to kill cells. And one of the things about it is when we talk about moist heat versus dry heat is that water actually makes cells more or the proteins more unstable, which means that you can use moist heat for less time at lower temperatures and have the same effect as dry heat at higher temperatures for much longer times. So that's why, like when you see the autoclaves or things like that, we tend to use moist heat because it is more effective at shorter periods of time. Just a couple notes on this. We talk about how resistant spores are. Spores generally need temperatures above boiling. So you'll see here on this chart that we tend to use about 120 degrees, 130 degrees of moist heat, and somewhere between 120 and 170 degrees for dry heat. So this will effectively take care of the spores. And to an extent viruses, because viruses tend to be more heat resistant. Viruses can be more susceptible to chemical controls, but in heat, viruses are more resistant. The last thing is, how do we determine these times that you see here? And this comes from the thermal death point or the thermal death time. What we do is we have organisms that we know are controls, and we'll place them in whatever situation we plan to do for this heat to see how long it takes. What we'll then do is we'll test for microbial death. Do you guys remember from the last topic how to test for this? Hopefully you do. But what you do is you take a sample and you put it in a nutrient broth, nutrient agar, and allow that sample to incubate and see if it grows. If it grows, then it means that you have effectively, or you have not effectively killed the bacteria. If it doesn't, or if it doesn't grow, then you have effectively killed. So that's how we determine what these temperatures are. So like for instance in lab, when we have you flame sterilize your loops, if you weren't to ever go to the inoculum and just streak out on a plate your blank loop, you would know that you had effectively sterilized. So that's how that works. And we use the same thing in autoclaves and a variety of other heat methodologies to make sure that we've reached that thermal death point. So our next mechanism is cold and desiccation. Now, these are not super effective controls, so they're not ones that we use too often, especially because cold doesn't even necessarily kill, it only slows the growth. In fact, this is how we store microbes. We store them in a freezer, usually with a bunch of chemicals to help them survive a little better, but they don't necessarily die. And some, like when we talk about the psychrophiles and the psychrotrophs, they actually grow still. So it's really important that you know that cold is not necessarily the answer. One aspect of this can be desiccation. This is where we keep things in a very dry environment where there's not very much water, and this can cause the microbes to dry up and die. And so desiccation is somewhat effective, but it's very difficult to create that kind of environment when we're talking like a hospital or clinical or laboratory setting. One important note on this is if we combine cold and desiccation, we actually have a process called lyophilization. And this is actually how we preserve microbes for vaccines or other things like that. And what we do is we dry them and keep them cold, and we preserve the microbes. So like if you're going to do a vaccine, you'll mix some water in with that before you give the vaccine. So if you're ever in a doctor's office or somewhere where they're giving vaccines, ask to see the bottle before they mix it with the water, and you'll see the lyophilized sample within there. Another effective method that we tend to use a lot besides heat is radiation. So if you remember from topic 1, gamma rays and X-rays are capable of sterilization, while UV rays are only capable of disinfection or disinfection. So We use UV rays a lot in laboratories, like in cell culture or anything like that, because it's readily available, it's not too damaging to surfaces, and is able to knock down the cell volume within a surface area or anything like that. We'll usually use it with countermeasures as well as alcohol or bleach or something else to help combat that disinfection rate to make it sure as low as possible. We also use this type of stuff, radiation, in fresh foods. So meats, things like that will be irradiated to stop the growth of microbes on them. So that's just another mechanism that we use radiation. Now, I talked about this in the first topic, how filtration is actually a mechanical method, but here for conciseness, we've just built it into the physical controls. And you should all be fairly familiar with what a filter is. We have some kind of fiber with some pores in it, and we'll run water or air through it to siphon out any of the microbes. Now, we can use a variety of surfaces and a variety of pore sizes depending on what we're trying to prevent from passing. And so this will work for a variety of things. Like if we want to have a liquid that we don't want to heat because it might break down some of the components in that liquid, then we can use a filter to filter out the microbes without damaging the solution. Now, we can't necessarily heat the air very effectively, so this is where we use like HEPA filters, and I'm sure you've heard of those before. And a HEPA filter is an air filter that will filter out the microbes. You'll see these on everything from vacuum cleaners to laboratory hoods, so they're used in a variety of applications. Our last physical control is osmotic pressure. And we've talked about this time and time again, is that even before we knew what microbes were, we were preserving foods with salt and sugar. We didn't know why it was so effective, but it was an easy way to keep things somewhat fresh. And so this creates a hypotonic solution or a hypotonic environment, causing plasmolysis in the vegetative cells. So the water will leave the cell, causing it to shrivel up within the cell wall and die. It's never a sterilizing technique, but is an effective technique to keep the microbial growth at bay. So that's osmotic pressure. So remember, and to review these again, just to make sure you have a good feel for them, we have heat, moist heat, dry heat. Moist heat being it could be boiling water, it could be a pressurized autoclave, things like that. Dry heat could be an oven. We have cold and desiccation. We have radiation, we have filtration, and we have osmotic pressure. So make sure you have a good handle on all these before you move on. Because as I said, you're going to be using these a lot in your professional career, so it's really important to understand them now and how they work so that you're able to master these techniques in the working environment. So this is the end of this topic lecture. Please let me know if you have any questions, and if not, I'll see you in class.